Hi, welcome to my channel. So for today our topic is Jga Apparatus, which has been repeated almost 6 times over the last 10 years in MUHS. Hope you like the video. So let's get started. पहले Jga Apparatus की definition क्या है? So when the DCT comes in contact with glomerulus of the same renal corpuscle, structural modifications occur in the DCT, efferent and efferent arteriole. तो structural modifications इनके cell में होने वाले हैं. The entire modified structure, tubular and vascular components. Tubular में हम DCT को include करते हैं and vascular components में efferent and efferent arteriole को. And the cells between them is called Juxtaglomerular Apparatus. So JGA के 3 components हो गए, एक tubular component, एक vascular component and the cells between them. So the JGA includes Macular Tensor of DCT, Jg cells which are modified cells of efferent arteriole and extraglomerular mesangial cells जो हमारे cells between them वाला component है, which is also called Les's cell. So let's see these in the diagram. So ये Jg apparatus का diagram है. This is taken from G.K. Pal. So in the DCT we have macular densa. In the efferent arteriole we have Jg cells, and the cells between them are the extraglomerular mesangial cells. So ये एक और diagram है. Like in case someone has a doubt like how the DCT comes near the glomerulus. So this is a practical diagram which properly shows that DCT and glomerulus come in contact, which is not usually evident in the normal diagrams we make. ये एक और diagram है. Deep blue color से we have extraglomerular mesangial cells which are also called Les's cell. In the distal tubule or DCT we have macula densa, and in the efferent arteriole we have these granular cells known as Jg cells. So now let's define each component properly. Now what is macula densa? So these are modified epithelial cells of DCT. These cells get modified when the DCT comes in contact with the efferent and efferent arterioles. What is the function of macula densa? So these act as a sensor that monitor change in ionic composition, change in flow of tubular fluid. So the macula densa monitors these things in the lumen of the tubule. So in case lumen में जो fluid है उसका ionic composition ज़्यादा हुआ या flow कम या ज़्यादा हुआ, then it sends a feedback signal to the renal corpuscle to change the rate of filtration so as to meet the need of the kidney function. So after receiving this feedback signal, the renal corpuscle can decide whether to constrict or dilate these efferent and efferent arterioles so as to bring back the original condition. Now let's define juxtaglomerular cells, which are also known as Pulson cells. These are granular epithelioid cells present mainly in the tunica media of efferent arteriole. Now what is tunica media? So this is a diagram of artery wall. So here we can see on the outermost layer of the artery is tunica externa, and just inside it we have tunica media. So these granular epithelioid cells are modified vascular smooth muscle cells with an epithelioid appearance. These are highly granular because they contain secretory granules. And what do they secrete? So they secrete renin, which activates renin-angiotensin system. Now let's move forward to our next topic, that is Les's cell, which are also known as extraglomerular mesangial cells. Now these are found in a triangular space. This triangle is formed above by macula densa of the DCT, on the left side by afferent arteriole, and on the right side by the efferent arteriole. So these are present inside this triangle. These are mesangial cells which are present outside the glomerulus. So they are also known as glomerular mesangial cells. These are agranular cells which secrete renin and erythropoietin, while the Jg cells were granular and they secreted only renin. So erythropoietin is an important point here. Now the functions of Jg apparatus is the Jg cells secrete renin, which activates renin-angiotensin system, which is involved in regulation of blood volume and blood pressure. Now Les's cell, which are also known as extraglomerular mesangial cells, secrete renin and erythropoietin. Jg cells were granular while Les's cell are agranular. Now the macular tensor act as sensor. They detect change in rate and volume of flow and also the ionic composition of the tubular fluid. They provide feedback signal to glomerulus to change the rate of filtration, and this forms the physiological basis of tubular glomerular feedback. From the tubule, the feedback will be given to the glomerulus, and thus they control glomerular filtration. So let's revise whatever we studied. These extraglomerular mesangial cells are present in a triangular space. The boundaries of this triangle are formed above by the macular tensor of DCT, on the left side by the efferent arteriole, and on the right side by the efferent arteriole. These are agranular and they secrete renin and erythropoietin. These macular tensor act as sensor which detect the rate of flow and the ionic composition of the tubular fluid, and thus they give feedback to the renal corpuscle to change the rate of filtration so that to bring the normal condition. While the Jg cells are granular and secrete renin, which activates renin angiotensin system. I hope you loved the video. You can consider subscribing to the channel for the latest updates.